So I have some other questions that are related to RLD that I want to put out in the questions. One of the questions that has come through chat is for the blinding. So, typically the question is how do you blind, the RLD, the appearance of the RLD is different and it's obvious, and how does one go about doing it? Turkus, we have a procedure, but I like to hear some opinions of the panel. I mean, it's pretty straightforward that you dispense it out into separate containers and label them A and B, and then give it to your analyst to do your tests, and that should be good enough. The other question that
you can even, just put duct tape or something like that, an opaque tape around if you don't want to take it out of the tubing. If your product, if you're dispensing it straight from the tubing, and as you'll see in some of the videos later, I think it's relatively straightforward to be able to blind. You're just looking at at A and B basically. Right.
The other question is what happens if when you're doing the comparison generic, for B studies, if what happens if RLD is not available anymore? How do you compare? That's a huge. And I have other questions regarding RLD of my own. Any suggestions? Anything that people can do when RLD is not available to develop a generic product? Tough question.
Yeah, it is a tough question, I think, and it happens not only in the generic world, it sometimes happens in the new drug 505(b)(2) application as well. So I think the problem is kind of same if RLD is not available. So as I mentioned before, we have to take a holistic approach. So it is not like one size fits all type of approach. So we have to see the entire scenario, and we want to first hear from the sponsor or the applicant what they propose, because they know their product and better than we do. And we as a start, we solicit what they are proposing. So sometimes if the RLD is an NDA product and it is not available, we want to see if there are other generic product approved based upon that RLD. So even the NDA product may not be available, but the generic. And then we go and check that Q1, Q2, how similar they are. And then we go for the Q3, how. So all this thing, basically, we want to see a complete story as a holistic approach, that what are the weapons we have to fight with. And we try to be as scientific, but at the same time as flexible as possible. We understand the sponsors' dilemma and concern about going through the whole clinical trials and all. So we try to address their concern and see what they have to propose to us. And when we are working in a regulatory agency, we most of the time, or always, we have to wear two hats. One is the scientific hat, other is the regulatory hat. So we see how we can breach the two and come up with a scientifically and regulatory science solution. Not always we can make the applicant happy, but we try our best. So my advice to the sponsor in that case will be, you propose a case, giving all the parts you have, what you have, and then we can evaluate. And if we can give you any suggestions what can be done, we'll definitely give to you. But yeah, the main bottom punchline is we consider each case on a case-by-case basis. So there is not one solution to every problem. Thank you.
Other question that has come through is what kind of product which product do you use for sensitivity, the test or reference? Sam?
Me? Okay. Others can comment too. So there's pros and cons. Usually the reference product is used when developing the method, when validating a method. There are instances, however, when the reference product is so expensive that the cost of using it in the validation becomes a bit oppressive. And so if you have a well-controlled, a test product that's manufactured in a way that's very well controlled, then you could use your test product in theory for the validation as well. Now, the reason I say pros and cons when you get to the sensitivity, selectivity, specificity part of the validation, the part when you're using the 50% and the 150% products, those are not going to be GMP batches of product. Those are going to be manufactured at bench scale. And so one of the things that can happen is that if you've got test product manufactured at the bench scale being compared with a reference product, or even the test product, manufactured under different conditions, then could there be some differences in terms of the relative response or the specificity? I think there could be. That's why there's still some allowance. That's why that's not 0.97, it's 0.95. But there's also different ways that you can produce that 50% strength and the 100% strength. One is you use the same base formulation, and you just adjust the amount of drug. So it's the same formulation, and I either put 5% drug, 10% drug, 15% drug. That's one way to do it. The other is that I say, well, I take my base formulation that has 10% drug, and now I take the one solvent, my dominant solvent, and I just exchange that out for more or less drug. So the way that you do this can also impact the way that things work, and it's actually helpful to describe in an application how you prepare those altered formulations. And for on these various ways, that it may be helpful to actually have data showing, if you'd like, that you have excellent proportionality of release when you actually use your test batches, because what you're assessing here is not the product. You're assessing the ability of your IVRT method to have consistent and proportional responses in response to the kind of standards that you're using in it.
I think I saw some question on the chat about, I think someone is insisting on discussing the aerosol product and also ophthalmic. So first of all, I want to clarify that ophthalmic is not within the scope of our discussion here. But the aerosol, if it is a topical aerosol, I have seen topical aerosol products. They have conducted IVRT, and I think Kailash also has some experience. I probably saw data coming from her company. So if you are successful in developing a method with the topical aerosol, and if you can quantitate like how many pushes you are using with the pump or the actuator, and only thing you have to make sure that you can put a thin layer on the membrane and let the foam break and put a thin layer. And if you are successful and if you can reproduce it, then it is doable. We have seen data in the agency who has used the topical aerosol and have successfully developed IVRT with the topical aerosol product.
Yeah, we have done that also. And generally, we start that off with first the bulk and then go on to the foam product. And that's doable. So yeah, the development aspects remain this. I mean, the considerations are all the same in developing IVRT method for foam versus the regular semi-solid product. So that's one thing I wanted. Another that probably hasn't come up through the chat, but something that has concerned me a great deal is the variability of the RLD, release of RLD, from lot to lot variability of RLD that we have seen many times. And let's say you develop and validate your IVRT method using test one lot of RLD, and then you for some reason either it's expired or run out of supplies and go and get another lot of RLD and do a pivotal comparison, and lo and behold, you fail. So that's because of the variability in RLD, not because of the variability of the test product. So how do we come around this situation? This is this requires a little bit more testing, thought-provoking. I like to ask panel members if they have any thoughts on that.
So I'll offer my thoughts. I think you got to really get to know the RLD. And that means understanding different batches of the RLD. And as part of doing that, to understand whether there's a trend. So why are you seeing variability? Is the variability because it's a very old product and the manufacturing process is not well controlled? You know, that's usually the first assumption, but maybe not necessarily the correct assumption. So before you kind of throw the RLD product under the bus, I think you also want to kind of understand, is there a trend as the product ages, which may happen with your product as well over time? And I think it's very helpful to characterize different lots of the RLD product at different ages in its shelf life, and to understand whether there are going to be implications for how you develop your own product, how your product is going to age, and how you want to do those comparisons.
Okay, any other input, feedback? No. Looking at the chat.
This is Margaret. I'm seeing so many questions about ophthalmic products. I just would like to make a comment, and this is my personal opinion. I'm not representing anybody here. When you go to ophthalmic products, you need to make a distinction if they are immediate release or extend release. If they are immediate release, it's going to be very difficult to do anything. An IVRT or IVPT for an immediate release is going to be very difficult because the product does not stay in the eye very long. So all your efforts should be concentrated in extend release products. And in general, extend release products are by injection or by a surgery. So if you talk to anybody about ophthalmics, they are going to make this distinction very clear, because as soon as you put anything on the top of the eye, it doesn't matter where, because there are several places even on the top of the eye that you can put the product. Your eye is going to make everything possible to remove that strange object that you put there, even if it's a simple eye drop. So when I'm seeing all these ophthalmic products, make it a distinction between if it's immediate release or extend. And remember that extend in general is a product that is applied by injection or by surgery. So there are lots of situations that we are not going to do anything because the product doesn't remain in the eye very long time. If you apply an ointment in your eye, it's removed in my case, is removed almost immediately. So why you are going to do all these evaluations for a product that does not remain even on the site of application enough time to do even some kind of action? So this is my personal comment, and I'm not representing anybody.
So allow me to say something that we've got an audience out there. Clearly, some of them are interested in ophthalmic products. And I think, I want to say something really, really clearly here, that we use the term IVRT, and we have to use it in context. In the context of everything discussed during this workshop, it's the IVRT used for the non-sterile topical semi-solid dosage forms. These are not for the ophthalmic products. The tests that are done that may be also called IVRT for ophthalmic products are fundamentally different tests. And so, I think while there may be some aspirations to have some kind of future area where there's maybe more alignment between that's outside the scope of this workshop. And I just want to be really clear, because we've seen confusion arising in recent years, since both situations are called IVRTs, but they are completely different in the way that the tests are done, the scientific principles, the data analysis. We're not really talking about the same thing. And I would just hate for someone to come away thinking that they're the same thing. They're completely different things.
Yeah, same thing with the injectable implants. I saw in the chat box someone asked question about injectable implants. So again, that is not the scope of this IVRT, but I can tell that it's a lot of echo. Anyway, so it's a different thing. You have to develop different method for that. Yes.
It's gone. Okay, we have just a couple, maybe hardly a minute, and I just want to pose one question about backflow, back diffusion of the receiving medium into the dosage form. This is a huge question, and the questions are asked: how do you avoid it? What do you do? How do you determine it? Perhaps we can address that in the next panel, unless anyone has a quick comment.
I agree to defer to the next panel.
Right.
And
Maybe almost.
if there's one last piece that I want to revert back to something that Sam had referenced regarding preparing your formulation to reach 150%. And by taking out the dominant solvent and adjusting it to account for the drug increase. Sometimes you are not able to attain 150% and maintain the same solubilization. And in that instance, what would be the approach? Would you maximize it at wherever you can get? If it's 120, 130%, move forward? Because I think you fundamentally change your formulation if you're having suspension or something else. If you just put in 150%, but it's not the same arrangement of matter that you had at the 100% strength.
It's a really good question. Now, this doesn't happen often, but it does happen because some of these reference products, they sort of maxed out what the formulation is designed to do. It's not designed to take an extra 50% of drug. And so when you try to do that, it really just doesn't work. So in those situations, there's a few things that can happen. One is, you might be able to change, let's say, micronize the drug and be able to actually develop that. And sometimes that can work out, other times it won't. What you may have to do is say, well, I can't go up to 150%. I can only go up to 130%. And it may be okay to be able to say, look, I did it at 130%, and look, I can still discriminate it. So I have a suitably discriminating method. So I know we've run out of time here, but I will acknowledge that that happens and that it's scientifically very challenging when that happens. And if that happens in the midst of something that you're dealing with, and you make every best effort scientifically to address the issue, and you say, look, this is a function of the product I'm working with, there's no way around it, have a discussion with the agency about it in a pre-ANDA product development meeting.
Great. Thanks. That's been my approach. Just wanted to get that quickly addressed. Thanks.
I will discuss that in my presentation, but yeah, the very last questions, and this is the question was asked, is that when you have 150% of the dose, comparing 100 and 150%, or even 50%, 150%, do we not change the molecular structure or arrangement of matter within the formulation? And what is responsible for changing the release rate? Is it the different arrangement in the matter, or is it the concentration of the API?
I hope it's dominantly the concentration of API.
We hope so too.
But why don't we pick this up in the afternoon? There'll be lots of good discussions there.